Colleen, do I have access to my screen share? It's not giving me, there we go. All right, hello everyone, and welcome to, actually I should say, leuk je te ontmoeten. Welcome to Late in Leuven, also known as Webinar Series Part 4, Stay on Target. We're going to talk today with 4 awesome people about therapeutic targets, but also how we are going along in our understanding of the pathogenesis of ALS, leading to the ability to clinically develop some of these targets. So without further ado, I've learned before, I need to stop showing my screen now. Hopefully everyone can see that. Now you see the wonderful panel in front of us. For the panel, can you see yourselves? It's good now on the screen? Yeah, okay, perfect, great. So I'd like to start by introducing the esteemed panel. First of all, we have a professor in the Department of Neuroscience at KU Leuven since the start of the year, Dr. Sandrine de Cruz. Previously, many of you will know, a very prolific set of publications on all aspects of ALS, working at the Ludwig Institute alongside Don Cleveland in San Diego. And Sandrine, previously to that, you were in Geneva, I believe. What led you to going to Don's lab and to starting a focus on ALS?
So indeed, thanks, David, first for the invitation and for the introduction. So yes, so it's about 15 years ago already that I got into ALS, and the reason is, one is actually 2 of the other member panels here, Pierre and Christine. So when I was doing my PhD, I was working on mitochondrial biology, and it was emerging at that time that mitochondria were playing a role in ALS, and I've been always fascinated by neurodegenerative disease, in particular ALS, being a devastating disease. And of course, pioneering work from Don Cleveland led me to going there for my postdoc to get really started on ALS.
Great. Well, thank you for being here. I think we have a similar story coming up on that. And actually now I guess we do have 3 mitochondrial specialists and 1 person who's got a lot of energy. So maybe this is the connection point to what we have today. So our next panelist is very well known to the ALS community. She's the Francis and Joseph Weinberg Professor of Neuroscience at Thomas Jefferson University, also the co-director of the Weinberg ALS Center in Philadelphia, and previously the scientific director of the Packard Center for ALS, which means she knows lots of cool things that we'll never get to see, that we'll see coming up. Dr. Piera Passanelli, thank you for being here.
Thank you.
Piera, you were in Utrecht, apparently. When I looked at it, it was interesting. You were in Utrecht, which is a big ALS area, but you weren't working on ALS. And then what led you to become the caspase expert with Bob Brown when you moved there for, I believe, the postdoc?
Yes, well, long time ago, actually. First of all, thank you, David, for the invitation. And it's very nice to be online and having this discussion with all of you. Hopefully we will be able to answer most of your questions. For me, it was much longer than Sandrine. Unfortunately, 25 years ago. I'm that old, and it was really serendipity. I was in Utrecht for my PhD doing really basic research on learning and memory. And after my PhD, when I moved to the United States, I wanted to work on a disease-related research. I didn't want to just do basic research. And so I applied to several laboratories, and Bob Brown called me. And at that time, I have to say, I never heard of the disease. And so for me, I just did some homework before my interview, and then the rest is history. And it was, I think, I've always said the lucky day of my life. And that was 25 years ago. Yeah.
Well, we're very lucky that's how it worked out, because it's hard to believe 25 years, but really appreciative to have you here. So our next panelist is also someone that a first name would give it away in the field. We have Dr. Ludo van den Bosch, professor at KU Leuven as well. 2 Belgian located individuals today, also the King of Leuven, been there for a very long time, I believe, at the Center for Brain and Disease Research, also a group leader there of a large group. And actually, I should say this to the audience out there, if you want review articles on many different aspects of ALS, this is the, Ludo's lab has got some amazing ones in recent times that cover a wide swath of pathogenesis. So highly recommended. Ludo, was it Wim Robert that pulled you into ALS originally?
Absolutely. I was about to leave academia when Wim Robrecht returned from the States, where he did a postdoc with Bob Brown. And I also think that Piera was there around the same time. And when he was looking for somebody to start his lab, he approached me, and I didn't have anything else to do, so I said yes. And I think it was one of the better decisions I took in my life. I hope my wife is not listening. So since then, I'm working on ALS, and I think it was the same as Piera. Before I was recruited, I had no experience in neuroscience, and I had also no clue what ALS was.
Well, another very lucky pull for the ALS community. And so actually Ludo is also the co-chair of the 2023 Gordon Conference on ALS, and he's the co-chair with our next panelist, Dr. Christine Vandevelde, who is a professor of neuroscience at University de Montréal CERCHUM, and probably one of the people I'm most indebted to in anywhere in the world, who I learn from on an everyday basis as a co-chair of our Scientific and Medical Advisory Council in Canada. And Christine, many of you will also know, had previously also been working in San Diego with Don Cleveland. And prior to that, you are a Winnipegger. And working with the late Arnold Greenberg. So was it the mitochondrial pathway for you as well to find your path to ALS?
It was actually, and up until this moment, I didn't realize the connection between us, but it actually, so my background was in apoptosis and cell death mechanisms, and they were focused on mitochondrial permeability transition and mitochondrially target BCL proteins. And in fact, it's actually Piera's papers on caspase that actually had me looking at ALS, because I wanted to take the basic biology and move to a disease. And it was actually, Piera, it's your papers that moved me towards ALS, and I went to Don. And then, because I'm from Winnipeg, I went to San Diego, and I was just obviously not going anywhere else. The day I visited San Diego, it was -30 in Winnipeg, and it was +30 Celsius in San Diego when I got off the plane. 60-degree difference in one day.
And yet you went back to Montreal?
Yeah, I know. I know.
Question mark. Okay. I'm not complaining. We're very lucky to have you in Montreal. Okay, so thank you all for being here. So these are just to preface for the panel. This was an idea, and we're very grateful to Biogen. So I'm not sure if I missed mentioning that off the top, but this is an ALS Canada discussion panel that is sponsored by Biogen, and they give us an opportunity to try something a little bit different. We, as anyone, there's a lot of webinars out there where you will get one person's opinion on things, and we thought that it might be nice to complement that with some discussion panels that's aimed at the scientists to really give some ideas and opinions from leaders in the field as to where we are in certain aspects of ALS. So this is the third one, and it's really starting to think about, where are we at in the pathogenesis of the disease. So before I wind you up and hopefully let you go with all those great thoughts, the idea for this one kind of came from the fact that it's incredibly exciting where we're at in ALS research, at least in my mind. You can start to see where the puzzle pieces are connecting. You've got nucleocytoplasmic transport, you've got mislocalized proteins. You have all kinds of issues with aggregation of proteins and things that need turnover, and you have problems with the turnover mechanisms. You have issues with transport of things to the processes, and then you need to have RNA and proteins in those processes to function properly. All of these different things that we can see easy connections from a high level at. But at the same time, if you go in and you read a review article, you can see there's lots of contradictory things. It's very complicated. I can point out even just like the C9 loss of function review article, Ludo, that you recently had. From a high level, I can think about the different things that might potentially be involved, but once you get into the details, it gets really complex. So with that in mind, and thinking about somebody who's maybe coming into the field or fairly new in the field or a trainee, I guess the question is, where do you feel we are at in our understanding of ALS pathogenesis? And do you feel that we have any more of these major landmarks to get through? Or do you think there's things on the horizon that are like the 1993 SOD1, and then the non-cell autonomous work in the early 2000s? And then there's the TDP43 discovery, which suddenly RNA was important. Then C9, which changed the world again, and then this age of gene discovery. What are your thoughts on whether or not we have the puzzle pieces we need and we need to put them together, or whether or not there are big pieces still out there? And I see Ludo shaking his head, so I'm going to ask him to start because maybe.
Thank you very much. No, I think as long as we cannot cure ALS, I'm convinced that we still are missing pieces, that we don't understand it completely. Having said that, I think that is the major problem in the ALS field. That is that we know quite a lot about what is happening in the different models that we are using, whether these are in vitro models, whether these are animal models, small animal models, rodent models. The major problem is that the observations that we see in these models don't translate so easily into the clinic. And I think that that gap is the most puzzling thing. Why is all the findings, all the cures that we have to cure some of the mouse models, why don't they work in the hospital? And I think that is because we are still missing something. And I'm pretty sure that we can discuss about what we are missing later on.
Okay. Is that echoed on the panel? Do you feel similar?
Well, I agree, but I would also say that there's also multiple, let's say, compounds or strategies that we tested in particularly in the mouse models, that we went further ahead, but actually the effect that we observed was often modest, but still we went ahead. Or, it was done on not enough numbers, and then later on realized, oh, actually there were nice studies out there and published that would have a certain number of animals to really be reliable. And I think this is also, that also has contributed. But I agree with Ludo that there's still much more that needs to be understood.
I agree with Sandrine, and I agree with Ludo also with the premise that until we know, I wouldn't go as far as cure. I would say until we really are able to transform ALS from a fatal disease to a chronic disease, and so to successfully treat whether ALS as a whole or the subgroup of the disease, I think the challenge is really the modeling the heterogeneity of the disease into our models, more than really in the ability to translate whatever therapeutic approach we have tried from the animal models to the patients. Because I think in that respect, I agree with Sandrine. I think we haven't listened to the animal model told us that those drugs were not translatable, if that's a term, into the clinic, and yet we went ahead and tested them. So whether we still have something ahead, like the next C9orf, the next non-cell autonomous concept, you name it, that's why we're doing research. I think I don't think we can say that we know most and we understood most, because technology will also give us the possibility, new technology. Until 5 years ago, we didn't have the iPSCs as a tool to really dissect even the sporadic forms of a disease. Now we do have them, with all the pros and the cons of the iPSCs, but as far as we are now technologically, it is as good as it gets as a window into the nervous system, right? So with the evolution of research and technology, perhaps we will have another breakthrough. We have to continue to do that, I think.
With all three of you and all of the comments you've made, and I would layer on that to build off of your comment, Pierre, that heterogeneity of the disease is something that is not well understood. And so we make mouse models looking for particular phenotypes that are mostly motor-based. And we've had an evolution of understanding of the disease that it's no longer just a neuromuscular, motor neuron-centered disease. But I don't think that we've necessarily, our definition of what is a good ALS model hasn't co-evolved. And so, but part of that requires that we need a better clinical understanding of the disease and sub-stratification or subtyping of patients, and then appreciating that we make models that address particular sets.
Can I follow up on Christine's point? I think you said something incredibly important, and that's why I think the research has to work very closely with the clinician and vice versa. Yeah. Think of what happened with the C9orf72 and the realization really that ALS is part of a spectrum of diseases that also include FTD. When 25 years ago, I started to work on ALS, the assumption from my colleagues, clinicians, right, was that, oh, ALS is a disease that is terrible, but it spares the mind, right? Okay. Yep. Now, in 2011, going back to the same clinicians, they will tell you, oh, but we've always saw that there was some executive function that was. Now, if we are not able to communicate with one another, right, those information are never going to be translated in real time. And I think that what I'm very proud, whether we are able to treat ALS or not, is that the ALS community in the past, in particular in the past 10 years, became so collaborative, not only like among all of us who do research, but also so collaborative with the interconnected, with the clinical folks, that I think this collaboration and working together can get us there a little bit faster. Think about it, the heterogeneity, right? We do see it in clinic every single day. Patients have different sites of onset. Patients have different disease progression, right? Patient can have FTD or not. And so now that we know how to clinically, that the patients are different, yes, they get the same diagnosis, I think we'll be able to model a little bit better in the subgroup, a little bit more precisely. So I think you said something very, very important, Christine.
I think the SOD1, discovery of the SOD1 gene for ALS as the first gene for ALS was both a blessing and a curse, because it conditioned the field to look uniquely for these, motor phenotypes in animal models, and blinded us, if you will, to these other symptoms that are known in the clinic, and doesn't allow us to really see that heterogeneity well.
I completely agree, but I also think that the mouse model is sometimes, and then I'm talking of course about the SOD1 mouse model, is sometimes blamed for the wrong things. I mean, it's not a bad mouse model at all. It's maybe not representing all aspects of ALS, but if you ask me, what do you expect from a good ALS mouse model, then I think you can tick all the boxes for the mutant SOD1 mouse model, more or less. I mean, there are of course some smaller things that upper motor neurons are not degenerating as prominent as you would expect. But then the question is, what's the reason that everything that works in that mouse model doesn't work or doesn't work as good as we had hoped in the clinic? So is that only heterogeneity, or is it that we are maybe too late when we start the treatment of the patients? Because in the mutant SOD1 mouse, and also in all other mouse models that we have, we can start treating quite early, even before these mice have symptoms. And maybe that is one of the reasons that we don't manage to translate what we see in the lab into something that cures ALS in the hospital.
Actually, I would love to go back to. Oh, sorry.
No,
I
go
was just
ahead.
thinking. Well, I was just thinking in terms of what Ludo just said, and jumping even to a point we were going to get to about models and you know, some people say, oh, this needs an IPS. Other people say, I don't trust the IPS. Some people say this has you know, you shouldn't do this in a TDP43 model because we don't get a motor phenotype in this particular model or whatever. Some people say you shouldn't do the G93A because it's not representative of a lot of the pathology. There's all that stuff. So I would love to hear more about how someone listening could, from your opinion, validate, or what they should do to probably best validate anything that they think is promising. But actually, one other piece to what Ludo just said is, if we are able to get to a point where we can pre-symptomatic, like we can earlier diagnose people earlier in the clinic, whether that is just getting people to tertiary clinics faster, probably a big piece of that, or even finding diagnostic biomarkers and an earlier program to be able to look for these. Do you think there's anything out there that we tested that we should retest? Because I guess there would be a question based on what was said, was something good tested and just didn't work because we didn't have the right clinical trial design and or earlier diagnosis? And do you think basic researchers would stand up for anything in particular that's been tested before and just maybe didn't get a fair shot?
I think a lot. I think a lot, to be honest. I think that there are many things that now are discarded as potential treatment strategies because they were not translated into a therapy in the best way possible. And the problem there is, once a clinical trial is negative, whether the trial was designed properly or not, it's almost impossible to put such a treatment strategy again on the list. So there is no funding for that kind of research. And so I think, well, another aspect, and I don't want to blame others for things that went wrong, but I think also target engagement is another important aspect that sometimes is not taken into account sufficiently. What works in a mouse, and maybe you can reach a target in a mouse, that does not mean automatically that you also reach the same target in a human, in a patient. These are the kind of things that I think can explain at least some of the failures, and yeah, I can go on like that for a while. I must say there are other aspects as well. But yeah, I think the question is, I mean, once a trial is negative, it's very difficult, I think, to bring it back to the table and to motivate or convince a company to try it in a different way.
I would like to echo what Ludo is saying in terms of pharmacodynamics and pharmacokinetics and target engagement studies. I don't want to cite some of our work, but I think we have clearly proven, using the SOD1 mouse with Riluzole, starting Riluzole treatment at time of onset, that nobody has ever done it, that way, and mind you, Riluzole really showed no effect in the mouse model, right? That there is this phenomenon that is called acquired pharmacoresistance in ALS that really prevents drugs from really reaching the target in the central nervous system, and this is disease driven, right? And unless we do block these drug transporters, we will always, even for drugs that are permeable to the blood-brain barrier, we will always be able to deliver the drug, but eventually the drug will be pumped out. And therefore, going back to your question, David, perhaps there are some drugs that show no effect in clinical trials that we should revisit. They were based on a solid scientific rationale, but we didn't do the proper studies, and we didn't really do our due diligence in studying this phenomenon of pharmacoresistance, right? The reason can be many. my experience with this is that eventually, I think we should really start to work a little bit more together, also engaging the pharmaceutical companies much sooner, right? Because each of us knows how to do only all what we are able to do. And the heterogeneity and the complex nature of this disease, I think, demands that all of us work in collaboration. We are all academics, and in our academic labs, we are able to study really the mechanism in our petri dish or in our animal models, and we are able to make models. However, we want to compare all these different models to get to the answer, but that's about it. We don't have the know-how, and actually we don't have the infrastructure, we don't have the money that the pharmaceutical company have to do drug development, therefore also the pharmacodynamic and the pharmacokinetic studies to validate all these models and then to translate to the clinic. I think that until we work with this mindset, we will always have this kind of disconnect. And therefore, model validation and translation into the patient will always be challenging.
But to just follow up on that, Pierre, I think you're absolutely right. I think, it's really encouraging times, because in the recent years we're seeing now these, how shall I say, this consortia happening where there's more and more funds put in having partnerships between academics and in industry or biotech. And I think that's indeed a key, so that there's more and more of those, so that, as you're saying, it's critical that we work more collaboratively, not only as academics, but also with biotechs, with the clinician, as we said before, but also interdisciplinary, I would say, with the genetics, because it's really all the pieces of the puzzle together and all our expertise together that at the end will make the difference. Otherwise, yeah, we're good at what we do, but we need to learn from the others.
Yeah. So, okay. So if we then, I guess that leads to one of the other questions that I had thought of, which was, based on what Pierre has just said and what was followed up on, do you think that there are probably some interesting therapies that were tested in the standard paper is hypothesis, proven, now I need something that I'm going to pull out an inhibitor or whatever, and I'm going to prove that it has an effect on motor neuron degeneration or glial activation or a phenotype of some sort. It gets published, and then it's there as a part of the abstract, usually at the bottom, and then goes away. Because as Pierre said, unless somebody has the ability to get the right connections to move that forward, it has proven its point for that particular paper and then kind of disappears into the ether. Do you think that there is any room in the field then to have sort of like a Eurovision drug screen thing, where it's like all of the winners from these various studies are then put through some sort of standardized way to figure out which ones might be real viable targets out there for the field to push forward as a potential clinical development piece? Or is that just too big and difficult?
I think what you described, Dave, is exactly the mission and mandate of the ALS Therapy Institute. The challenge there, though, is that they have restricted themselves to validation in only the G93A SOD1 model. So the concept exists, and what they're at this idea of taking the literature, they comb the literature for potential hits, and then they test it in large cohorts. That's wonderful, but it would be so much better and so much more powerful if we would branch out to include additional models, because maybe the target is not relevant all the time in the D93A mouse model. Maybe it'll be more relevant in a C9 model or in a TDP mouse model. But I don't know that I would, bet my life that I could say that this is of the 25 or 30 TDP mouse models that are out there, that this is the one that should be used to expand on. So I understand the challenge. It's not to slam ALS TBI. I think that there their concept is smart, but I think we're limited that we don't have. It comes back to we don't have the models that necessarily reflect well what is the clinical heterogeneity in the patients.
I think that what you are describing, I agree with Christine, but what you were describing, David, perhaps doesn't exist yet, but we are closer to get to that point. For instance, Target ALS is doing some of that work already, right? Not perhaps at the large scale as it would be required, but one has to start somewhere, right? And perhaps even going back to the mouse validation. Now, the discussion started to think about are the IPS better than the mouse model? And again, it really depends what we use the mouse model for. If we are talking about drug development, perhaps the SOD1 model is a good model, not to study efficacy.
Right.
If we all agree that perhaps the drug screening can be effectively done in the iPSCs, then the SOD1 mouse model or whatever other model, one single model, can be used to study in vivo, which is a requirement to then go to the patient, drug-drug interaction, pharmacokinetic, pharmacodynamics, right? And then go to the patient, perhaps. I think that level of coordination, more than the LSTDI, has started through Target ALS, but association like yours and every other association in the world can play a big role in doing that standardization and putting us all together, I think. Obviously, we need to agree on biomarkers, on targets, but I think we can do that, even with solid discussions that science is driven by discussion, and I can agree or can disagree, but the data then at the end are the data, right? I think a coordination in that way. So I think your question is incredibly important, David. The question is, how can we get there?
And I also think we should collaborate more in the sense that we should focus with more than one lab on a certain target. But I'm sometimes a bit afraid that yeah, every lab is focused on one mechanism, on one target, and that is then the one that is pushed into the direction of a clinical trial. What I would be much more convinced if others can reproduce the data in a different model or even in the same model before you take the very big step to start treating patients. And I know it's not the kind of work we find easy money for. I mean, just reproducing somebody else's work. I mean, you don't get it published in a good journal. If it doesn't work, you don't even make friends. I mean, that's another consequence. But I think on the long run, sometimes the translation into something that is used in the clinic is just going too fast. I mean, I'm also sometimes, and I don't know whether I hope to at least have some controversy here. I also sometimes see some trials starting in the hospital where I have no clue on which scientific basis that was started. So of course we all know the trials for which there is very good evidence, the antisense oligos. There I think nobody doubts that that's the way to go. But then in addition, when you look at the list of trials that are ongoing, there are, I'm not gonna give a number, but there are a large number of trials where you have no clue what was actually the basis for that trial, and hopefully there is a good basis for that. And I think, well, by just being a bit more critical sometimes, yeah, a lot of frustration and a lot of disappointment and a lot of money can be saved. But I don't know whether the others share the same opinion.
I think there's another, yes, I definitely do. I think the other aspect too is that for some of those trials where you're not really sure of the scientific rationale for them, you end up burning patients into that trial, and now they're not available for maybe some other trial that has a stronger rationale. And so there's sort of an overall, it's a domino effect in general. I mean, there can be.
Yeah, and it's driven by other things than science. Not clear what exactly is driving that. I'm not involved in these decisions.
And given false hopes, and that's giving false hopes for patients, and that's not also, that's something that is critical that we need to take into consideration, unfortunately, right? So that there's quite a few where the basic science, the light science is not there. We don't know if it's there. At least, as you said,
it's not public. Yeah, it's not public. It could. I always think, okay, maybe the company has done its homework, but it's not published in a journal. And despite that, it's pushed forward, and patients, yeah, they are recruited. There's a lot of money spent for that. And in the end, I mean, we as scientists can already more or less predict, I think, that the chance that it will work is quite low.
Yeah.
And then I think some clinicians say, yeah, but it's better than nothing. I'm not so sure, actually.
I agree with you. I think it's always a very fine balance because the desperation that this disease brings, we are not clinicians, and we don't have to deal on a daily basis with patients who really, need some hope, right? And that's why I think that this is a combination of things. Perhaps one thing that we could do as scientists, really, to the benefit of the community, is really starting to have this honest discussion with the patients, right? Rather than saying, oh, we would have predicted this is not working, right? And saying why this is not working and why up until now, perhaps the science has not been able to identify the right therapeutic targets. For all the reasons that we just said, right? So ALS is heterogeneous. The other reason I always think is that I think there are three challenges that we have, not only with ALS, but all the neurodegenerative diseases, that I think that if we are able to convey our challenges in research to the patient, then we can really serve the community much better. And then we won't rush all these clinical trials. One is, again, the heterogeneity of the disease. The other is that up until recently, we could never do what the cancer field has done to harvest the actual tissue on a living patient, right? So we are still not doing that. So when the cancer field became successful is when they understood that one cancer was not the same as the other cancer, right? But by protocol, they can do it. We are still not doing it. We will never be able to do it. But at least now with the iPSCs, we can at least, whether I'm not advocating for the iPSCs, but what I'm saying is that we can at least have a model that is as closer to the patient and to the actual tissue, right? In terms of genomics, metabolomics, whatever omics we think about. The third challenge that I think we really have to be able to, that we will always have, is that contrary to the cancer field, we need to resuscitate something that is dead or is dying. And here it goes to an early diagnosis, here it goes to the early intervention.
Yep.
It's much easier to, once you identify the target, to kill a cancer cell. So far, nobody has been able to resuscitate anything, right? So I think we have to balance all these challenges, and sometimes even if we don't have all the data, we have to go into a clinical trial try.
So keeping something alive is more difficult than killing something. That's your point.
Right?
Mm-hmm.
I agree.
Actually, your comment on cancer brings up this is an aside piece, but I remember there was a description of a movie with Sean Connery back in the 80s, Medicine Man or something, where he goes into the jungle to find the cure for cancer. And then I saw a comment below from somebody that said, which one? which cancer did you find the cure for in the jungle? Because I think at the time, exactly that, it was the cure for cancer, not the heterogeneity aspect. But you've all really provided the perfect segue here for the next set of questions, which is really diving into, where it is exciting. And because I think what Ludo was referring to a lot is there's a lot of generic therapies that are considered to be either anti-inflammatory, neuroprotective, repurposed because a company has that already. They already are making money off it in a different indication, so they can go back and try it in ALS. And we're in a situation in the world where as trials become more and more available, and as we move a lot of these things out of the lab and into the clinic, there's going to be a limited capacity of early stage ALS patients to put in these trials, and we're going to have to start being selective as to what's going in and what things you say, no, that's probably not the best one. So with that in mind, for the panel, I guess the question I have is, okay, so actually I should just say like that the Target ALS is a great example that when they described last year the things that they were investing in, that's exciting stuff. Like you're talking about going after LLPS, the Staffman story, obviously, the HDAC6, the P3BP1, but these are things that are actually focused on something in the lab that is ALS pathogenesis focused from a number of different groups, kind of confirming that it has some role in ALS. So those are where it gets really exciting about potentially clinically developing these. Aside from the ones that you all might be closely invested in, what are the other things or pathways that you think are most exciting as potential targets that we need to figure out how to sort of get in that same pathway?
Nobody wants to reveal what.
Nobody wants. Just
if I would know, I would not tell you. Is that the correct answer?
Okay, that's fair. That's fair.
For me, my hesitation is because that's such a difficult question to answer.
Yeah.
It is a difficult question.
I agree.
It is a very difficult question.
I'll start with one pathway that though it comes back to that I'm not sure we understand enough of the biology, but one thing I'm super intrigued about is what is the connection between viruses and ALS initiation? Is there something there that we're missing? But it comes back. So antivirals, I think, could be very interesting. I understand there's some preliminary work happening in that domain with the retroviruses, but maybe it's not just retroviruses, maybe it's other viruses, because I think that plugs into several of these. Obviously I've been playing in stress granules for the last few years, but it's not just stress granules. It plays into RNA metabolism, since various viruses like to co-opt cellular, host machinery for various purposes. So it's one that I think, though, but which one, I don't know. I just think that we should be studying more about this. We should try to understand that more. And it's not necessarily just in the lab, it's understanding from patients. Like, we don't really have a lot of epidemiology studies for ALS, which I always found strikingly odd. Because there's such a huge environmental component that we don't understand, and that viruses might be part of that environmental component. I mean, I think it's got to be almost undoubtedly, probably, yeah, but we don't have the data for that. There's a little bit of data out there, but
you want
not
to
a lot.
apply for some COVID money?
Well, I don't know about your place, but here they're handing it out by the buckets full.
I hope that there's no link with ALS, because then we have a problem in Belgium.
The whole world is a problem.
I agree with Christine. I thought my idea was also to mention retroviruses and whatever. There are now not only papers, but also companies, I think, that they are looking into the development of some of these therapies. Another thing that I think we can still explore, and I don't have a candidate there, one example could be ataxia, is really the genetic modifiers that I don't think either by using an unbiased screen with the yeast, ataxin-2, or the flies, or a combination of both, and then validate immediately the presence of those genetic modifiers into ALS patients like they've done. There is, I think, something, these unbiased screens can really bring us new potential targets, and the ataxin-2 right now is that I hear that they are going to start the phase 1, phase 2 trials, Biogen soon. I think that that's really what excites me. Now, if we want to learn from the Alzheimer field and the beta 42, perhaps clearing these TDP43 aggregates is not going to help, right? Perhaps it is true that TDP43 is, to cite a very famous person, is pathobabble, right? But we won't know until we do the actual trial, right? But I think that the rationale for testing and using the antisense against ataxin-2 is very solid. And so that's why this genetic modifiers field can really, together with the retroviruses, are something that excites me right now.
So yeah, I was about to say the same. So just about the modifier, and this is key, putting more effort there, understanding, because of the heterogeneity and emphasizing on the genetics. There's so much genetic data sets available out there, and digging more through those along with also these genetic screens. I think this is critical, and that will really help us understand better what's different in this person versus the other, and then do more personalized in a way medicine if possible.
It actually brings up too what in what Ludo said earlier about trials that have gone by, and thinking about Michael Van Es is doing the lithium carbonate again, but with UNC13A mutation individuals, by going back and looking retrospectively at who might have been genetic responders based on a specific thing. So maybe there is a pathway to look at old trials, but as long as the data was solidly collected. And what you're saying, obviously, with you know, the ongoing ataxin-2 study, and then the potential to really focus in on this. Okay, so devil's advocate on that then. If, let's say, the antiretroviral therapy, let's say that's a significant component in a large proportion of people with ALS, or I gathered maybe Christine was talking about an upstream trigger as one of the stages. And someone would say, like a lay individual might say, well, then why are you looking into liquid-liquid phase transitions? Why are you looking into stress granules? All these very complex things when there's low-hanging fruit out there. Like if SOD1 was the common denominator between the first neuron that has pathology and the rest of the neurons where it transmitted, should the whole field just be putting all their eggs in that basket because the rest of it is moot? So with the virus, the same question would be, how does somebody new to the field, so let me position this, someone new to the field, a postdoc who comes in reading their first review articles and says, well, if retroviruses are reactivated and a significant part of triggering ALS pathogenesis, why don't we just aggressively all go at the viruses because the rest of it won't matter? So how do you rectify that for somebody? Is it's basically the safety net, because if it doesn't work, we need to have other things, or? Because
that requires early diagnosis. And that's what we don't have. We don't have markers for early diagnosis. And that's why I think, personally, I think mechanisms that are focused a little bit downstream, and one of the targets I got left out in the previous question, and I apologize to local colleagues, the neuromuscular junction. So mechanisms that maintain neuromuscular junction and the dynamicism that we now know that we didn't know before, although it's funny because the clinicians knew it, but the mouse people didn't, and how to incorporate that. And so if we had early diagnosis, then yeah, we could work upstream and just, you know, target virus and that'd be fine, but we don't have that necessarily. Like, so these things have to co-evolve together, because there's going to be patients that will not have that early diagnostic. And so things that can stabilize the NMJs, I think that's super important. And in fact, for a field that is originally described as being solely motor neuron, it's always sort of surprised me of how few groups are focused on the neuromuscular junction, which is the business end, or at least
I think this panel is maybe an exception.
But we have been
maybe this group is an exception.
Yeah,
not representative of the field.
No, it's encouraging, right, to see people like you, the guys that are actually now focusing on that, because I think that that's where we need to be, because that's what's going to have clinically meaningful impact, to borrow a famous phrase recently, is to actually stabilize the neuromuscular junctions. That's going to give patients actual real clinical benefit, and going to make
to induce reinnervation, which, I mean,
but that'll have actual real tangible results for patients. They can still button their own shirt, for example.
Yeah, I will even
improve quality of life. Go ahead.
No, go ahead, Sandrine, please.
No, I was just adding, yes, indeed. I think it's also looking into molecules or strategies that improve quality of life of a patient, and targeting the neuromuscular junction is also one of those.
And for people who are coming new in the field, I would really even go further than that. I think we tend to focus on one thing. It is either the pathway or the ear or the gene of the ear, right? And that's wrong, because we keep saying that ALS is a heterogeneous disease. So there is maybe a role for the stress granules, there may be a role for the RNA metabolism, there is still a role for SOD, because we like it or not. We still didn't treat patients with the SOD1 mutation, right? There's still a role for the TDP43 pathology and you name it, right? Retroviruses and everything else. We really tend to forget yet another gene, because perhaps it's not so common, or perhaps because right now the field is going. Listen, you started by talking about non-cell autonomous disease. Do we really care anymore about what C9 does in glia? No, right? We are all still focusing on C9, and C9 seems to be the most important form of ALS. Yes, it is the most common. Whether it is the most important, do we know it?
Right.
Right. If I was another patient,
not when you live in China, no.
That's a good point. But
it's like, just to build off of Pierre's point, though, what I think she's saying is the field is sort of the swarm of bees that moves to the next flower upon whatever came out in the next big paper. And it leaves exposed many interesting targets or pathways that are sort of half solved, if at best. But all of it in the end is going to come together to give us that, a full perspective of how these things interact. And many of these pathways are going to converge, they're going to cross. And we see that already for the really, really rare familial ALS variants. You see a bunch of them that they start to stack up in a pathway, and you're like, oh, okay, like I'm thinking TDK1 optineurin, BCP, you start to see it. And so I think we don't have all the puzzle pieces just yet.
And I think that as it happened, I don't know, I want to share, I don't know what happened to you when you guys went to Wim or to Don, or I went to Bob, but people who come to work with that, we may be biased at this point. We not biased in a bad way, right? But we know what we know, and our research, even though it expanded over the years, started with caspases and mitochondria, and I'm not working on that anymore, right? And I'm working on FAST. We still perhaps you are doing the same, all of us, right? Neuromuscular junction, this kind of thing. But we know what we know, and then there may be a postdoc, there may be a student who comes from a completely different background, that is looking from a different perspective that we haven't looked at. And perhaps that's the new field to go, right? And so I think there is room for everybody, which also drives down the competition that sometimes really kills rigorous science. If we only were a little bit more open-minded, I think also in the expectation of our research. I don't want to be philosophical, but I really think that being a researcher needs to be open-minded. I don't need to only look under the microscope what I'm looking for.
I agree. I always tell my students, I always end up doing something I know nothing about. And that's good for me, and it's good for science, because I'll never ever know everything. And to keep those blinders off and not necessarily follow the swarm of bees all the time.
Yes, so that's actually, oh, sorry, Ludo, did you want to?
No.
Okay.
I just wanted to add, never a dull moment. So we have already been working on so many different pathways. Yeah, well, it would have been, of course, not as much fun if we had to stick to the mitochondria from the start. Having said that, we are focusing on mitochondria again, but anyway. History repeats itself.
Yeah, actually.
And I also think the other point I wanted also to make, maybe we sound all a little bit too negative about things. We should also stress that that the only way to solve the whole problem is to recruit even more talented young people to start working on ALS and to motivate them to try even better and harder than what we already did in the past. So I think that's important. We should not sound as old men, in my case, having tried so many different things, having failed so many times. I think, well, I must say that I also learned a lot during all these years.
Why do you say that we failed? That you failed many times? I mean, I'm one of those.
In translating the findings into something useful for patients. That's what I mean by fail. Not in getting insights into mechanisms in a motor neuron in a cell. There, I think we were very successful. Genetics, I think, is extremely successful, was extremely successful, but I'm not a geneticist, so I cannot benefit from that. But I think, well, cell biology, I mean, we learned a lot about motor neurons, no discussion about that. When I say failed, I just mean, well, I would love to retire sooner or later, knowing that I made a difference for ALS patients. And that didn't happen yet. I'm still hopeful, but it didn't happen yet. So that's why I said fail. No, don't misunderstand.
But I agree with you that we shouldn't be, can I say it publicly, party poopers, but because in reality, listen, Riluzole and Radicava, they may do little, but they do something. And as my husband says, when I spend money, he says it's the sum that makes the total, right? One after the other, perhaps. And I think that we have failed only when we don't learn, only when we don't have this discussion, and only when we design a grant, when we design a project in our lab, we don't take into account those failures, right? In reality, we are having this discussion precisely because of that. Because listen, if we knew what we were doing, it would have been, and what we were looking for, we wouldn't call it research, right? If we had all the answers, why would we do what we are doing, right?
Yeah,
so
absolutely.
I think that there is a failure only when you say, okay, we are done. Until we keep trying, and we keep trying always better, guys, maybe there is the answer out there already somewhere. I don't think we have to
No, I don't want to sound too negative. And well, when you start writing a grant, you never start by saying I want to cure ALS. It's always much more focused, much more specific, and well, I think we reached already some of our goals. But yeah, well, of course, working in a field where there is a disease where people die from in such a terrible way, I think you cannot be blind for the fact that in the end you spend also a lot of charity money. Yeah, now I'm looking to David again. So this is money from patients, from families, from people that really want us to make a difference for patients, and that's maybe less than what we hoped for. But yeah, I'm repeating myself. So, but I'm still young, so I'm still gonna work hard on that.
Ludo, you and I are not young anymore.
It all depends what you call young.
And you are older than me, my dear. So
you feel and look young. Is that all that matters?
Yes, I feel young.
Yeah.
Well said.
Yes.
I have a provocative follow-up question, and then I have a less provocative follow-up question. So the first one is, okay, so if there's a swarm of bees, and let's say that the last 7 years, the swarm of bees has been C9, a large swarm of the bees. Now, obviously, there's the individual swarms of bees each year, as Piera said. Like right now, it's probably C gas is the thing at the moment, sting for the moment, for this set of months. But okay, so C9, there are a number of very interesting therapeutic targeting mechanisms that are being clinically developed now, or in the early stages of it, from small molecules to addressing, obviously the ASO is already well into phase 1 with Biogen, but all of these different unique ways to target it. Do you think that we should almost, thinking from a health parity standpoint or whatever, this is a very dangerous question, but should there be prioritization away from C9 now if we think we've got that as a subset of people who were sort of got some strong things to test over the next several years, but yet heterogeneity wise, maybe the TBK optoneurin autophagy, the inflammatory response pieces, maybe the RNA biology pieces where the swarm should go. But that would obviously make a lot of people have to shift what they do because there's a lot of C9 work.
Yeah, but I think I'm not so sure whether that's the case, because what I tell also, well, people in my lab is try to find a niche and don't go where all the others go, because what is the chance that we will be able to compete with the good groups in the States and in Canada? So we need to go for something, yeah, where nobody else has been looking, and I think that's then the challenge. But maybe from the outside, yeah, it can give the idea that people indeed focus too much on one aspect. I also think that, well, when you start working on something new, it's easier to be successful than when you, for instance, want to go for the next big thing in, let's say, a SOT1 model, because already so much has been tried. So the chance that the next strategy that you are using is the one that will work is not so, yeah, it's not so high. When you go and work on a C9 model, whether it's an iPSC or a mouse, but I don't think there is a good C9 mouse model, then yeah, the chance that you will find something new is much higher. So I think from that point of view, from a scientific point of view, I think there is always some kind of incentive to go for something completely new, even if we don't understand what's going on with let's say, SOT1. I mean, we still don't know what's actually going wrong there, but we more or less gave up. If you now send in an application for a grant panel, the chance that you will get money for a SOT1 project is not as high as for a C9 project. And I'm very, well, careful in the way I'm saying it. I think it's it is what it is, and I think people, yeah, like new things, like new hypotheses, and this is all, I mean, a self-fulfilling prophecy. So yeah, in grant panels, we well, the same people that are doing the research are also evaluating the science. So in the end, but I don't think it's a good thing. I think we should give also money, and we should also give room for ideas that at first sight are not the hype of the moment.
Yeah.
But yeah, but yeah, it's not always easy.
Because they're grounded in good foundational science. I totally agree. I think that there's lots to be learned from sort of the projects that are a little bit outside of the main thrust of the field. I think there's lots to be learned. And sometimes also they challenge the dogma that the main thrust of the field is putting forward.
I guess it depends who you are, right, in terms of foundations and or NIH. From the scientific standpoint, I think that we still don't know not only what the C9orf repeats do, but C9orf itself does. So from the pure scientific standpoint, I think there is still a lot that we want to learn. From your question, because there are already developing a lot of therapeutic strategies, so perhaps one thing that we still don't know in C9 is what is that makes patients with the same mutation. Some of them develop FTD, some of them develop ALS, right? Because perhaps in approaching it therapeutically, we really have to go targeted, even in this subgroup, homogeneous group of patients, right? So, and then the same thing is valid for why do we have to focus only on C9 when there are still other ALS patients from which we don't even know the cause yet, right? Which is the majority, the sporadic and this kind of thing. So from the basic standpoint, scientific standpoint, I think we should be focusing on SOD, on C9, on FAS, on sporadic, because we still don't know. when we go to translation, I think your question is very important because of what you do, right? And I think if we go to NIH, we shouldn't have that bias, I think. When we review NIH grants, we shouldn't have that bias, and we should really review the science if it is not translational, right? Everything else that we do that we want to bring to the clinic, we should be a little bit more mindful. And saying how relevant, you mentioned the stress granules, right? And I work on that as well. So, but at the end of the day, how relevant for the intellectual standpoint is very, very important. Is it really an area of focus for the translation into therapies? I don't know. I don't know the answer to that, but
it's too early, I think. It's too, I mean, we should also not, I think, well, be too much biased by the fact that we should only do those things that are immediately translatable to the clinic, because then you just should stop and you should just start testing drugs. And hopefully there is industry that is doing that. That's not my goal is to understand what's going wrong.
I agree.
And yeah, hopefully that can then be used to develop something that helps a patient. And then the question is indeed, yeah, well, do you only have to focus on what is popular at this moment, which is, I think, stress granules and nucleocytoplasmic transport? And yeah, well, I mean, it's interesting and it's intriguing, and there is mislocalization of protein, and there is a connection between stress granules and nucleocytoplasmic transport defects. So, I mean, in the end, it all comes together. So I think it's still interesting, and we are still working on it. So.
Yeah, I mean, sorry, go ahead.
No, I was just wanted to follow on some what Pierre was saying. I think it's also important to follow up on having solid fundamental science, really understanding. We're talking about, okay, this gene or this gene, I'm talking about the familial forms, but this gene that we know, this gene that we know, but then even within that specific mutations, because what we're learning, for me, it's puzzling why a mutation is going to lead to ALS, why a mutation is going to lead to FT, or even another disease, yet for TBK1, there's examples like that. And so really understanding, but the basic science and doing and collaborating with those in vitro, but even in silico, I mean, there's so much that we can learn from each other, really to understand why this one does this one does that, and then to be able to translate. But there's still a lot of work, like in the field, I would say, battlefield, really to understand these basic mechanisms first. And that I don't think.
One example is even MATRIN3. Right? Is such a rare mutation for ALS, but it also causes a pure muscle disease, mutation in MATRIN3, and this could open really so many lines of research that have been not explored. And by the way, going back to the neuromuscular junction, it is made of those two cells, right?
Yeah.
So.
But you're right. But the thing is, actually, there's quite a few examples when we go through, right? You're right, Matrin, TBK, not to make a list, but VCPs and other. I mean, there's quite a few, even fast. There's some examples where mutations don't lead to ALS, but to other. Right. So, and I don't think we're exploring enough that, and we should and collaborate not just within our ALS community, but even outside from other diseases as well.
We need more money and more people.
Yeah, too many avenues to explore. And actually, I really appreciate the framing of all of this too, because I think that this is actually going to be really valuable if we allow it to, like, if you all are okay with it being streamed, because I think for the patient community as well, to see the passion, but also all of these things are going to be really exciting to potentially look at the validation of whether or not they can be clinically developed. So I think not going to the quick wins and just immediately saying this has to be translated, understanding it, making sure these are really good targets is going to be super important to making sure we have some things that are going to have a real clinical effect, as opposed to marginal things that we really can't decipher whether it's real or not. So actually, I did have a less. Oh, sorry, Christine.
If I may, I think that I agree with what you're saying, but I think we cannot underestimate the urgency that patients feel, and that they feel that they don't have time to wait for the basic research to be solved. And so it's partly on us to help them come to the realization of the value of the basic science, but the community needs to do that as a whole. And I think we are each responsible for what we put in our press releases and what we allow our institutions to say about our studies, because in some places things can get pumped up a little bit because they can then point to it as a donor hook and then use that to drive donor dollars into a local program, which is, you know, wonderful that there are people that want to do that, but we have to be, it's each of us have a personal responsibility to be careful about what we say our study says and what it does not say, and to work hard to convey what is the true outcome of any of our studies, and try, because it's hard, to ask people to understand that and have value in the basic science. And I can't imagine what a family who's affected with ALS, how they might be feeling that urgency, and you know, they see
Yes.
this press release and that press release, and in this age of misinformation that can happen so rapidly, as we've witnessed in the United States for the last few years. Sorry, but when you think about the misinformation that can happen and what can be spread so quickly across multiple borders, that somebody in some country on the other side of the world has had some, you know, anecdotal success with a particular therapy, and how it can completely catch like wildfire. And it only, I think, I'm sorry to say, it's quite a strong opinion. I'm sorry, I'm not. It completely undermines basic research that the field is trying to move real targets forward.
Yeah, that's a great jumping off point, actually.
You asked for that opinion, but you got it.
No, but that's a
For
great
free.
jumping off point too for tofersen being something that comes from years of understanding the potential of, or how, oh, you can't hear?
Yes, yes,
How
yes.
basic research. Sorry, go ahead.
Christine is
Bruno.
gone. No.
Oh.
I think Christine has problems. Can you hear us, Christine?
Interesting.
No, she
We
lost
hear
connection.
you.
Yeah, we can hear you.
We can hear you.
Sorry, I don't
Yes,
hear anything.
loud and clear.
That might be my connection, sorry.
Yeah.
Well, just know that when I'm talking, it's not important. So as long as you can get it before other people speak, you're fine.
She said something about fake news, so maybe that's why she's now muted.
She got shut off. Okay, so while Christine works that out, I actually think that that's a really important point. And so a couple points made there that I think are really important too. It's certainly not to undermine when there's proper messaging, the drugs that are in the clinical pipeline give potential hope for something for people, even if they're not rooted on strong science to begin with. This is something that is there, and as long as properly messaged and you know, ethically all handled, then this is a great, you know, the urgency for people in the community. Certainly, I would never want to have undersold how important that is. And tofersen's a great example of something that comes rooted from basic science and ALS specifically, all the way through to what looks really exciting for 2021. So I actually do have a quick tofersen question for all of you, but before that, I want to go back to the non-controversial question I had, and it's just sort of like a curiosity thing. So heterogeneity being a huge piece here across the board with what we've been saying, and you've got a number of these studies across the world, Answer ALS being a huge one, Ambrosia in the UK, you've got some that have been connected to NCALs, you've got SALSA in Australia. They're going to come up with large data sets, and it's going to be up to the individual when these are made public through data browsers to really be able to interpret that. Do you think there will be some necessity for a really strong dissemination of what that means as people start to understand the data, so that the average basic researcher in another who maybe can't decipher all that information can start to understand how their stuff works into the heterogeneity of ALS? Because that's going to be huge amounts of data. It's going to be hard to understand what it means to be able to move your stuff towards the clinic, will it not?
Yes, and I think that probably one step, there should be thought now about how to recruit in groups that are experts in data mining and machine learning consortia around that, that bridges basic science, scientists like myself, with how to utilize those large data sets. I know there are some groups out there, but are there enough? I don't know.
Right.
Yeah, and it's a bit magic, eh? I mean, but now I sound very old-fashioned, I suppose. The bigger the data set, the more there is hope that something will come out. But how it will come out, for me, I mean, I don't have the right background, I think. And yeah, the strategies that we have now, for the moment, they didn't come out of these large data set, data mining, artificial intelligence, but that's maybe the future. I mean, I don't want to say that it's not a possibility. What I know is that all these kind of new technologies are extremely expensive, and that's then becoming a problem. The amount of money, I think, that is going to Project Mind, to answer ALS, to all the other initiatives, it's a huge amount. And I hope that it will be well invested, which I'm still waiting for to see that, but there's still time to find out. But yeah, well,
I agree. I really don't know. It's really above my intelligence, actually, to be able to decode.
We are too old. We are too old for that, Piera. We are too old for that.
But I think that the purpose of those collection is it's sort of what the genetics has done for the familial forms, right? And here is the genetic with more data and more complicated pathways, right? Is what the genetics has done for the familial form. Now, they are just hypothesis originating data. They are not the answer for sure. No. They are just the beginning, right? Yes. I believe that it will be up to all of us to look at this data and really cherry pick, like we have done before, our pathway of choice based on our expertise. And with the help of artificial intelligence, absolutely, because I don't really know how to read those things, right? But then eventually we will have so many pathways, and we will go to the pathways that we really know how to study, and and then perhaps we will identify the starting point.
So, because what I'm thinking in terms of, so you're right. I think most of it will be cherry picking what people are looking for in the data, and then if it matches with the hypothesis, then you can move it forward. But I guess another thinking is, if focusing on human ALS is a huge frontier to evolve where we are with our clinical, like our basic research understanding and the heterogeneity of the disease. If you have, say, Answer ALS, which is doing iPS cells at a certain time point, and it's going to give something, a readout of something. You've got another initiative in Australia, another one in Europe. If you start to find where they are like, wow, even though this is read at different times with different patients, there's some harmony. Someone comes in and says, look, I can see this analysis is completely congruent with these other two. Then you start to say, someone can decipher that for everyone to say, this is actually important because we've looked at 10,000 patients with ALS, and they're all showing these 5 signatures separated or something. Is there, you think, value in that? Because I wouldn't, there's potential for those to sit there and not be mined purely because of what you're saying, that people can't understand. Too big for our human brains to comprehend it all.
I don't know if even the collection of these cells is done so that they are definitely not my brain and not the human brain. Maybe the artificial intelligence can do that. But for instance, the Answer ALS data are really comparing fast versus slow progressor, right? Now, I don't know what the Australian will do. Are they looking at bulbar versus limb onset? And even the way these cells have been made, right? Is it their standardization yet in the way we make these motor neurons, we make these cortical neurons? Is it there a thorough clinical phenotype? Are they FTD or pure ALS? And we don't know that. So how can we compare this data eventually? And perhaps there is a way to do it. I'm not a statistician. I'm not very smart, I have to say. So I really don't know. But
that's for the next generation, Piera.
exactly. So there you go. I told you that we are old.
It's for the next generation.
I think
Well, it's still
Sandrine
early
touched
days
on it.
as well, right? No, yeah,
Sorry?
I had a.
I was like, Sandrine touched on it earlier. It's this, you know, we need this interdisciplinary, intersectorial type research. We need to de-silo, and we need to see these data as hypothesis generating, not as things that we can validate our favorite hypothesis against.
Exactly.
Like that's a very different, let the data talk to us rather than us asking the data a particular question and saying, do you agree or not agree?
Yeah. So the first time when we had a large data set, I think it was proteomics, we went to somebody who's going to help us, and the first question he asked us, what do you expect? Yeah, okay.
What do you want us to find?
Yes, that was exactly the question. So I said, yeah, that's what we don't know. I said, then I can't help you. Okay. It's already a few years ago, so maybe things have improved. So
well, that's actually one of the points that I had put down to bring up, but it's very controversial thing is, but maybe you can just do it with a yes and a no. Do you think there are avenues that we explore in ALS research that are not real? Are rabbit holes that are not actually valuable rabbit holes?
Yes.
You don't have to say which ones. Do you think they are?
All I'm going to say is yes, but yes, I do.
Yeah. And so
I
guess one of the other questions is, how do we start to shut off some of the rabbit holes, and where do we even begin, and how and who can do that? Because if you think about a limited funding environment in ALS, if some of it's going to things that we don't, as a field, collectively believe are correct, well, then is that a good or a bad thing?
Yeah.
Dangerous territory, I understand.
No, I think you have to be in a way naive and believe in the self-correcting power of science. I think, well, I'm pretty sure that, well, some things are maybe not correct, but not even done deliberately wrong. I mean, it's not every experiment is designed in the best way, and you sometimes get results that that are too good to be true, and then usually you are doing something wrong. But and that's not always your fault. I mean, you only realize it afterwards, and maybe sometimes too late. But I don't think you can avoid that. I think the only way to avoid it is stop doing research. Then you don't make any mistake anymore. And I don't believe, and that's maybe that people make these kind of mistakes deliberately. I think that are really the exceptions, the very, very, very rare exceptions in the ALS field. And I think also overall, I'm still a believer in the fact that scientists are fair people. And I think we all make mistakes, we all make misjudgments. I'm pretty sure about that. As long as it's not done deliberately, as long as you don't start inventing data points that are not there, or removing data, I think then we're talking about something completely different. But I think what you are referring to are hypotheses that on the long run don't hold, and where you have spent a lot of money to prove or disprove them. I think that's part of the game. That's part of research. Unfortunately.
So maybe the wisdom there is that if something seems too good to be true, test it again just to make sure. And don't
by somebody else. That's my first reaction. If somebody comes to me with data that are too good to be true, I'm trying to find somebody else in the lab that has to do exactly the same. And most of the time it works, I must say.
It reminds me of a quote from Heather Durham that I always remember, but she said we should always be aiming to prove our hypothesis false. That's what we should always have that in mind whenever we're doing stuff. And I agree with you, Ludo, that the cases of people that deliberately do something wrong, I think those are incredibly rare.
Yes.
There are those mistakes that are done by omission. So there are lines of research that have been launched that are, unfortunately, the background rationale or the appreciation of the broader literature hasn't been done sufficiently, and they only realize it way down the road that, oh yeah, you know, and so then all of that time that's been lost on,
or that you forgot to do the right control. I mean, sometimes, I mean,
it happens.
Yeah, it's and you know, and afterwards, it's always easy to say, yeah, you should have done it differently, but it's yeah, well, that's the problem. I always tell my people that they only should do the experiments that give the right results and not all the others. But that's the problem. You never know which are the right experiments. So that's the.
And but I agree. I never knew that quote from Heather Dorn, but it's very important because I always say that why don't we publish negative results? They are results.
Yes,
right?
Important.
Because it's difficult to get them published.
Yeah, but that's wrong because they are results. An hypothesis, by definition, is not a dogma.
Yeah. Yeah, and also it would prevent having others, you know, working again on the same and then finding that same negative result again. And so there again, it's wasted. And just for even that purpose, it's very important, I think, that we can publish negative results. They're very important. Yeah.
Someone has to revolutionize the scientific supportive environment for good hypotheses proven wrong, right? I guess. Anyways, I appreciate that you brought it all back. See, Ludo, you're not pessimistic because you brought it back to a very optimistic point, which is really that science is doing everything it can as to work itself out, and that, you know, in general, everyone is trying their best to get to these answers to ultimately get things into the clinic, into patients.
Absolutely. And we're only humans, so I mean, we can just do our best and work even harder than before.
So there's only 2 minutes left, and so I'm not going to ask that last controversial question. So you're off the hook for that, but maybe offline with everyone anyways, another time. Anything anyone would like to add to the discussion that, you know, if someone's out there and they're watching and they're finding their way in terms of understanding ALS, or just, you know, working forward on it, anything with respect to the discussion that's been said today? No? Okay. Well, for me, it's been incredibly
we need the young researchers. I tell them not to give up. And I think we also have to, this is a different conversation, but I think that the young people coming up, sometimes they see academia as not as a rewarding a career as maybe we feel it is, because the challenges of academia are getting more and more difficult. We also shouldn't undervalue, I think, trainees that are not necessarily interested in an academic research path, and put more value, and that we need smart scientists that are in all parts of the scientific ecosystem. So that
smarter than we are.
I agree.
That's true. Yep.
I totally agree.
And as Ludo said, we need a lot more great people in this field, because there's a lot to do and a lot to figure out, but we're on a good path. So thank you all so much for your very valuable time. Really appreciated. I could have done this for a long time, so I'm sure that all of you are glad that we're not going for another 3 hours. But thank you very much. Really appreciate it. Hope that other people out there listening will join us next week for the 5th of 5 of these, which will be focused with some clinicians talking about clinical trials and how we can improve those, as well as what's in the clinical development pipeline. So thank you all once again, and thank you to Biogen for supporting this and this idea, and hope everyone has a wonderful rest of the day.
Thank you.
Thanks for late Leuven.
Bye.
Bye.